Okay, it's started. Okay, everyone. Good afternoon, or good evening, or good morning, whatever you may be. It's a wonderful pleasure to introduce the speakers. Today's speakers, Domagoj Vucic, who is a great, long-time friend and part of this course since the very beginning. Domagoj is a senior fellow. I was going to say Department of Early Discover Biochemistry, but now I just realized that he's at the Department of Immunology at Genentech, a company belonging to Roche, and I'm sure everybody know about it. His position is equivalent to the full professor in academia. And at Genentech, Domagoj leads efforts to develop compounds that target RIP kinases, the IAP ubiquitin ligases for blocking and controlling inflammatory responses and or enhancement of the survival of damaged cells and tissue. So since very early in Domagoj's scientific career, he was interested he was interested in translating basic findings, signaling principles into novel therapeutic for treatment of cancers and inflammatory diseases. That passion allowed him to progress IAP antagonists from his initial mechanistic discoveries all the way to the clinical trials. And because of that great adventure, I just heard that recently Domagoj considered himself a cell death cowboy, man. I want to hear about that. I really want to hear about this.
Some
Okay.
other time.
Okay. So anyways, so because of time restraints, I will cut this introduction short and pass the microphone to Domagoj. Domagoj, super thanks again for being part of this.
Great. Thank you, Karina and Gu, for inviting me. Happy to be here. I would like this to be interactive, so please interrupt. And then I divided this talk in 3 sections, so I will make a break after every section to allow questions. And then we can formally have question time at the end. Okay. So, let's see. You heard over these 2 days a lot about cell death, and I just gonna briefly go over, obvious things that when cells die, the cells shrink, chromatin compacts, DNA is cleaved, and then you have apoptotic bodies that engulf that, and then they are eaten by macrophages. And so that's why apoptosis is not considered to be very inflammatory. But nevertheless, it is crucial for life because during the embryogenesis, tissues are formed and lost, and human body generates a lot of cells every year that needs to be disposed of, and most of that, especially developmental and immune cell death, happens through the process of apoptosis. However, deregulation of apoptosis can have pathologic consequences because if you have too little, then you can have autoimmunity cancer, but you have too much, then you have degenerative disease and tissue damage. Okay. So the kind of schematically that looks like this. You have cells that live in this layer, very happy. However, when they receive a signal to die, they detach from the other cells, and then you see chromatin condensation, blending of the membranes, and then eventually this chromatin piece engulf in the nuclear fragments, and that they would really be problematic for the body. But then macrophages come and they eat these cell fragments in apoptotic bodies and provide nice and healthy environment for the rest of the cells in that setting. Okay? And so when we look at that on molecular level, we have a couple of pathways that regulate apoptosis. So there is extrinsic pathway or death receptor pathway. And so here, TNF, for example, binds to TNF receptor 1, aggregates the complex, TRAD and FAD and so forth are bound here and leads to active caspase 8 recruitment activation, actually in 2-step process. but fast ligand or trail bind to their receptors and then directly activate fat caspase 8 and lead to active caspase 8. On the other hand, intrinsic pathway, it really leads to cell death upon treatment with chemotherapeutic reagents, irradiation, and so forth, sometimes even growth factor starvation. And mitochondria are critical here because you have mitochondria leaking factors like cytochrome C that then can activate APAF1, and then caspase 9 is recruited in the complex called apoptosome, and that leads to activation of caspase 9. That's to regulate that BCL2 family members that are critical part of this process that block this reaction, most of them. Okay. Then both caspase 9, caspase 8 can lead to activation of caspase 3 and 7, and then they cleave hundreds of thousands of cellular substrates and leads to apoptotic cell death. And so that's such important process is regulated at several levels. One level is on BCL2 level that regulates mitochondrial pathway. The other one is the on a caspase and death receptor level. So here, CIPs can, for example, block caspase activity, or XIP can directly inhibit caspase 9 or 3 or 7. And then you have amplification of the cell death process by leaking a mitochondrial factor, second mitochondrial activator of caspases, which is SMAC, because cytochrome C is the first one, that binds to IPs and prevents them from inhibiting caspases. Okay. So today I'm going to talk about how therapeutic opportunity can be found here. What is the options for targeting this cell death pathway to help patients? And I'm going to start with a BCL2 family, and then I'm going to go to IPs, mostly in context of cancer, and then I'm going to end with RIP1 and TNF signaling in context of inflammatory diseases. So first is going to be of cancer, and there is this well-known review, Weinberg and Hanahan, that describe critical properties of cancer. Some of those properties are, they are very proliferative, evade growth suppressors, and they can metastasize, go to the other parts. They induce angiogenesis to get this blood flow and nutrients, and the other one is resisting cell death. So that's one of those critical factors in that case. And so among that, BCL2 is the key factor because it was really the first one identified. Even in 1984, was identified as site of chromosomal translocation, which is characteristic of follicular lymphomas. Okay? Occurs like this. And then from 18 to 14, David Vo, now at Weihai, later found that unlike other oncogenes that were known in the past, that RAS and so forth, they simulate proliferation. This oncogene promoted cellular survival rather than proliferation, okay? And so there's a completely different genetic control, and that was really paradigm shift, that it's not just proliferation, but also enhanced survival can lead to cancer development and progression. BCL2 is frequently upregulated in variety of tumors, CLS, MCL, and so forth. And so these translocations cause increased levels of BCL2, and then that increases survival of the cells. And BCL2 family is also very complex. There are several members. So what I mentioned so far is BCL2 itself. They have these BCL2 homology motifs, BH motifs, okay? And so some of them are critical for interacting among each other. Some of them are indicative of the cell death, but pro-survival factors all have BH4 motif, unlike the others. And that's BCL2, as well as BCLX, MCL1, and also A1 BCLW. Then we have proepitotic family members, BAX and BAC, that are critical for execution of this process. And then we have BH3 only proteins that are critical for interaction. A lot of them, big so far, they are the instigators. They activate the whole process, okay? And these large members are associated with the membranes, whereas the small ones, mitochondrial or cellular, they can come to mitochondria. There are various ways how they can be activated. For example, some of them are stimulated by DNA damage. Hypoxia leads to p53-driven PUMA and OXA expression. Okay, some of them are driven by various stimuli such as taurosporin steroid or cytokine deprivation, bad BIM. Some by various metabolic processes, ion efflux, Taxol, oncogenesis. Some of them also by metabolic process, and some actually activated by death receptors because caspase 8 cleaves them, and only then that can inhibit pro-survival cell death. So there are various mechanisms. As you can imagine, human body is very complex, so there are also complex ways of activating these processes that involve transcription, sequestration, moving from one part of the cell to another part of the cell, phosphorylation, like bad, and proteolysis, such as a case with BIT. Okay? And when we think about targeting here, we have to understand that cells want to survive. So they have all these different family members pro-survival, and it's hard to kill the cell if you target just one of two of them. For example, BAD will target this group, BCL2, XL, and W, but not MCL1. NOXA, only MCL1. And as you can see, they by themselves will not cause cell death. But BEAM and PUMA, they can take all of them out, then they can by themselves activate cell death. And all that requires this BH3 motif that binds into the groove on the anti-apoptotic proteins, and that's how it inactivates them and allows cell death to proceed. Okay, so schematically, you can think about it in two ways. One is that you have death signal, and then this BH3 only protein can then attack BCL2 family members and prevent them from inhibiting BAX and BAC. But also there are some of them that can directly activate these processes. Either way, although this sequestration is more prominent, you will lead to conformational change in BAC and BAX, oligomerization, and then that can lead to mitochondrial outer membrane permeabilization, leakage of cytochrome C, caspase activation, and apoptosis. Okay, so this is the basic paradigm of how to target. And so now the idea is to get small molecule that's going to disrupt this interaction and then lead to downstream signaling, and in BAC and BAX dependent fashion activate caspases and lead to apoptosis. So idea was that in cancers they have these pro-survival proteins. How can we treat them with these BH3-like compounds and activate death of cancer cells? Now, the challenge here is that this is not a kinase, or this is not a protease that you have the active site. And so then it will be much easier because you know there are 2 or 3 residues. You have to go with a very small compound and you can block this. This is a much bigger region. And here, drug is meant to block protein-protein interaction. Okay? It's not meant to block enzymatic activity, but to block protein-protein interaction in this case. Okay? And so that there is this amphipathic BH3 helix that goes into this groove and binds here. So idea is how to mimic that in a minimal fashion. Again, structure of the protein was critical to understand how this whole family interacts, behaves, and how to target these proteins with the small molecules. Okay? So initially, there was company Abbott that developed compound called navitoclax, or ABD263, that was mimicking BAD. Okay, this is BAD is one of the BH3 protein. And in this fashion, it was blocking BCL2, BCLX, and BCLW. Okay, it was pretty large for the small molecule because there is something called Lipinski rules of 5, and one of the postulates is that molecular weight should be less than 500. This one is almost double, so it required a lot of work from chemists and pharmaceutical colleagues to develop this properly. You know? However, this was the first idea that you can really develop something like that even in 90s, late 90s. And although it didn't block MCL1 or A1, it did show nice activity and blocked BCLX, BCL2, and BCLW. Okay. Furthermore, the navitoclax was taken to clinic and showed nice efficacy, because CLL tumors expressed BCL2 and BIM, and if you disrupt that, you can prime cells to die. And there was efficacy already phase 1 in this resistant refractory CLL, 35% response rate, and progression-free survival was moved, and all that can be here. This is I show you reference here, so you can go read yourself and look at this very impressive inhibition of the effect and blockage in the lymph nodes and lymphocytes. However, there was one major problem with navitoclax, and that is that it was causing platelet loss, and dramatic one, okay? And that seems to be all depending on BCLX. The work done in David Huang lab in Weihai in Australia, published here, and some others really show that platelets don't depend on BCL2, MCL1, IAPs. They really want BCLX. And if you target BCLX cell, you lose platelets rapidly. That was done in mice, in other organisms, and here you see significant loss as you increase the dose. Or here in time, imagine within an hour and a half, you lose almost all of your platelets when you target with compound that will inhibit BCLX cell. Okay, so that's this thrombocytopenia was very severe. There are attempts at that time to maybe slow down the process or treat intermittently, but there are a lot of patients that had problems with that, and that was thought to be dose-limiting toxicity of navitoclax, and presented very big hurdle, And so then, if you want to have drug that you're going to treat patients for a longer time as in cancer, it has to have better safety profile, okay? And so then, folks in Abbott, and also at that time we collaborated Genentech with them, and in Weihai realized targeting BCL2 does not present the same issue, okay? And if you target BCL2, you can have efficacy in leukemias and lymphomas. BCLXL is actually critical for solid tumors, but also for platelets. So in this way, if you limit yourself and only target BCL2, you will not maybe target solid tumors, but only leukemias and lymphomas, but you'll be safe and you will not cause harm to patients in addition to what they already have to deal with cancer, okay? And so based on that, it was the idea to develop BCL2 selective reagent, okay, that will have efficacy in hematologic malignancies, okay? You will not hit BCLXL, so you will not have thrombocytopenia, and will get, if you get even better BCL2 efficacy and specificity, you will have better therapeutic window, higher exposure, and better effect in BCL2 dependent malignancies. And so that's the idea. Unfortunately, solid tumors then are not affected, but at least you can have a safer way of treating leukemias and lymphomas. Okay? So again, structure was critical to compare how BCL2 structure looks and BCLX structure work, to look at the differences, and there are very few outlined here, and that will only 4 residues are really different here. So these 4 residues are key to how to find selectivity, to find a way to bind to them and not to the in BCL2, not to this one in BCLX, and that way have a BCL2 selectivity and avoid hopefully a toxicity of targeting BCLX. And so then there was a lot of manipulation, a lot of combinations had to be done here, and modifications of this compound, and you basically had to lose some of these groups and modify them, and then eventually we got a little by little to lose a BCLX affinity and maybe reduce somewhat, but then you go step forward towards BCL2 selective. And then that was venetoclax, which is really selective BCL2 inhibitor. It has high BCL2 affinity, lower affinity for BCLX and MCL1, for BCLX desired, orally bioavailable, so it can be taken as a pill, and very active in BCL2 dependent tumor lines and in vivo and in vitro. And so you can see here a difference between navitoclax and venetoclax. Okay, so dramatic improvement here, and BCLX also you see dramatic improvement, thousands of fold increase specificity for BCL2 over BCLX in this case. And so these are the differences that make that possible based on those structural information. Now, venetoclax was tested in variety of animal models. Here in mouse xenograft models of lymphomas and leukemias. This was done in combination with rituximab, CD20 targeting antibody. You can see in this model, single agent shown nice activity, but when you combine them, really complete inhibition of tumor. In this case, now in different model, again, very moderate single agent activity. When you combine them, very long and very nice effect. Even 2 months after you stop dosing, you still see protective effect in this case. So that's very impressive. And based on that, and based on the safety profile, this was advanced then to clinical trials. And nice part about this compound, venetoclax, is that you know that you need to have BCL2 present. If so, it's you take the blood from these patients, you examine their cancer blood cells, you look at the BCL2 levels. If it's high, a very likely compound will work. Right? Importantly, when you look at this CLS samples ex vivo, you see very nice activity of venetoclax, twice improved over navitoclax. So BCL2 selective agent was better in this case in efficacy. And then in human platelets, there was really a huge effect and much better profile. And so as navitoclax was really reducing platelets, with the venetoclax BCL2 targeting, that wasn't the case. And then here with some clinical activity, when we see the lymphocyte count was reduced in majority of the patients, and then we see the lymph node size, we see this can also reduced bone marrow infiltration, all that really remarkable responses in overall response rate in these patients. It's very favorable, and safety was favorable, and based on that, venetoclax was approved for CLL and now other cancers that have BCL2 expression. So in 2016, FDA granted accelerated approval. Later, it was approved in Canada, and little after that in Europe. It was also pronounced, I think, year after, the most successful small molecule drug in the world for that year. And there are clinical evaluation ongoing in other tumors, and there are more now approved. That's what I told you. But it's one of the most successful drugs, and the only one so far approved that's based solely on cell death pathway. So cell death mechanism is the critical part here to do this. And we have drug cannot help all the patients, and that's because MCL1 is a major factor of resistance. Because when MCL1 is expressed highly, that allows survival of these cancer cells, and venetoclax does not inhibit MCL1. But and there is ongoing research. There are ongoing efforts to target MCL1. Unfortunately, there are a lot of toxicities because MCL1 is critical for many factors and normal body operation. And so therefore, it's very difficult to target it. Idea is actually to even reduce the levels of MCL1 with some degraders, but not fully degrade it, but just reduce the levels, that would already help in combination with venetoclax and Venclexta and help even more patients. Okay. So this is the part about BCL2. I will move on now to the other sections. Maybe I can stop here for a minute and give students opportunity to ask questions if there are any.
Anybody has questions? Normally, Domagoj, we were waiting to the final part
Okay.
and then do the big discussion, but I just see Giovanna already raised her hand. So go ahead, Giovanna.
I have a question about Batoclax, because I know it was developed after Venetoclax. Are you going to talk about it, or?
Which one? I'm sorry, I didn't understand. Which drug are you asking about?
Oh, Batoclax, mesylage.
No, I'm not covering this. I thought I cover just this one that was approved today, but there are
Okay.
other reagents. Yeah.
Okay, because that's a drug that I'm working with, and I was interested in asking some questions, but it's okay.
All right. Sorry. Okay. So the other way to think about targeting, in addition to venetoclax, as we said, a selective BCL2 inhibitor, platelet sparing, and so forth. There are ways to target cell death with proapoptotic death receptor agonist antibodies and proapoptotic SMAC mimetics. Okay. I will not talk about death receptor agonists today. I will talk about proapoptotic SMAC mimetics. Okay. So IP family of proteins are called inhibitors of apoptosis. They block apoptosis induced by both death receptors and chemotherapeutics. Okay? They are expressed in many cancer types. There are 8 family members, and they are characterized by BIR domain baculovirus IP repeat motif. That's a protein-protein interaction motif, and also helps to inhibit caspases. There are also ubiquitin ligases, and they have this ring motif, some of them at the very end, and that allows them to ubiquitinate a variety of different proteins. The reason we were also evolutionary conserved, just like BCL2 family proteins are found in C. elegans, but IAPs are not. In insects such as Drosophila, there are IAP proteins, but BCL2 family does not play such a prominent role. The reason why we were interested in this family is because of their also expression in cancer. For example, MLIP stands for melanoma IAP, very strongly expressed in melanomas, but non-normal cells, even benign nevus, almost no expression, and very strong expression in melanomas. Okay. So here's the paradigm for targeting IP. So we talked about BCL2, and this is now we're going step down in the pathway below BCL2 pathway. So BCL2 proteins regulate mitochondria, and then when cytochrome C leaks, and then you have activation of caspases, then IAPs would block caspases and inhibit, for example, XIP can block caspase 9 and block caspase 3 and 7 by direct inhibition and interaction, okay? And so now, when you have stimuli such as ionizing radiation, chemotherapeutic agents, and other factors, then another factor, in addition to cytochrome C leaks, SMAC or Diablo, and has the region that will allow it to bind to IAPs, and therefore takes them away from caspases, and in a sense weaponizes caspases. And now they're ready, fully active proteases, and ready to cleave a lot of cellular proteins and substrates, and therefore cause apoptosis. And so the idea is to have IP antagonists, to antagonize IPs with small mimetic, this small molecule that will have IP binding motif. Okay. And so similarly, as I said, XIP BR3 region will bind caspase 9, but SMAC has similar region, okay? And when SMAC binds to IP, then you have active caspases, okay? Again, remarkable evolutionary conservation here, because this heat, grim, reaper, and sickle, these are the proteins from the fruit fly, from Drosophila. And then SMAC and HTRA2 are human proteins and is the same residues, AVPI, or very similar. And then caspase 9 also has these similar residues. So you can see how SMAC peptide can replace caspase 9 peptide if it's provided in excess, and therefore cause cell death. Again, here we understand the structure, and we solve the structure of the BR domain of MLIP to understand how the peptide binds into this groove, okay, and then how we can mimic that. And so IP antagonists really mimic this SMAC protein, and they antagonize IP proteins, bind IPs with a high affinity, and block interaction with caspases. And as I said, all started with a peptide, and then through a lot of structure-based evolution of peptide mimetic, get a compound that has much better properties. So in this case, any screen of millions of compounds would not be better than what nature provided in form of this SMAC peptide. And that was foundation for all the companies that were doing this. Okay? Eventually, we developed, and the others, a lot of these compounds, and I'll show you just some example. For example, subcutaneous, a lung model, and you can dose this compound daily, and you see nice reduction. Here is a vehicle, and here is a compound. Very nice reduction of the tumor growth. In this case, for example, you see vehicle is here. This is control compound that doesn't work so well. And then there's another Genentech compound, a remarkable efficacy in these breast cancer cells. And then we can also go to orthotopic model. So we can go with ejecting lung cancer cells in the lung of the mice. As you can see here, pre-treatment. And after 2 weeks, a tumor grows very large. However, if you treat with a IP antagonist, great reduction of this tumor size. Okay. So based on this data, there are a lot of companies that advanced various compounds to the cancer trials that reached phase 1 or phase 2 for some of them. Okay. And critical part in the whole thing is, how do you know that your compound worked? How do you know that your compound really inhibited IP proteins? So while we were studying that, we realized that IP antagonists cause degradation of CIP1. And this is monovalent compound, this is bivalent compound, okay? And they do that in cell lines, or we can do that in vivo. Also, treat mice with xenograft, and we see a reduction of tumors 8 hours after treatment. And now we see that all of these compounds actually show in patients also this process of degradation of CIP1. So I will digress a little bit now, talk a little bit about ubiquitination, how that fits in the whole play here with testing reagents and validating their activity in patient samples. Okay? So for that matter, ubiquitination is also very conserved process where you have E1 ubiquitin activating enzyme, then allows E2 to be charged with ubiquitin, and then a conjugating enzyme, and then E3 is a ligases, then allow E2 to transfer or directly transfer ubiquitin to substrate. And substrate can be either K48 ubiquitinate, and then leads to proteasomal degradation, or that be 63 or 11 linear, and that leads to complex assembly and downstream signal, right? So in this case, we can have either a CIP1 can ubiquitin RIP1 and TRAF2, and that will lead to NF-κB activation, or it can be K48 ubiquitinate itself or NIC, and that will lead to proteasomal degradation. And so now, how does CIP1 ubiquitinate itself to cause this degradation when IP antagonists bind? Again, the critical part here was to solve the structure of the protein. And so we have here BR3 going to UBA, ubiquitin binding domain, CAR domain, and then ring. What you can see here is that protein kept in the inhibited form, where the ring domain is kept in check by BR3 domain. And only after a compound binds, it opens it up, and now you have active protein and ring domain that can very rapidly ubiquitinate substrates, including itself. Okay, so that this is this region. So basically, schematically shown here that you have ring is inhibited in this form, but antagonist binding allows opening of this conformation, and now dimerization. And so active dimer will now promote ubiquitination and degradation. And we have made series of mutants, for example, to show that. You see CIP1, if you look at this first lane by itself, it's fine, protein is here. However, if you put IP antagonist, is degraded. If you include proteasomal inhibitor, it's reduced. However, in this case, if you break this interaction, you open the protein, and right away it's already active, and it's already ready to be ubiquitinated and degraded, as you see in these images. So schematically, this works out like this. So TNF binds to TNFR1 and then recruits the whole complex. CIP1 is there. Then IP antagonist bind in this case, and what's going to happen is going to activate ubiquitin ligase activity of CIP1. As a result of that, you will have activation of NF-kappa B, but also proteasomal degradation of CIP1. When you activate NF-kappa B here, then you will make TNF, and then TNF can come in autocrine or paracrine fashion and activate this signaling. However, CIPs are not there anymore because they are degraded, and therefore you would lead to cell death and apoptosis in this case, because there is no more inhibitor to block that process. Okay, and that's how whole postulate is for how IP antagonists work and exert their activity in this case. And it's not just IPs that are inhibited in this fashion, because there are a lot of proteins that are ubiquitinate during apoptosis signaling. For example, in addition to IPs, MCL1 is often ubiquitinated and degraded. FLIP is ubiquitinated. Caspases can be ubiquitinated. Even some other adapter proteins, a lot of them can be ubiquitinated, and that serves a role to fine-tune and regulate apoptotic signaling in this case. Okay? Now, what's interesting that by looking at the cancer and IAPs and how they regulate the signaling, we actually uncovered the true endogenous physiological role of these proteins, which is regulation of non-canonical NF-kappa B. Okay, because what happens is that a non-canonical NF-kappa B is regulated by NIC, NF-kappa B inducing kinase. Okay, and there is a complex of TRAF3, TRAF2, and CIPs, and in that complex, NIC is constantly ubiquitinated by K48 ubiquitination and proteasomally degraded. So you can see here, you see NIC here constantly degraded. It's never there. However, if you put IP antagonist, then CIP1 is going to be degraded, and then NIC is upregulated, and then you have P100 processing and activation of this process. Okay. Okay, so now when CIPs are gone, okay, CIP is degraded, now NIC is stabilized, and now you have phosphorylation of IKK alpha, P100, partial degradation to P52, and activation of non-canonical NF-kappa B and stimulation of inflammatory cytokine release. Okay, and so this is not just for IP antagonist, because CD40 ligand or tweak that binds to FN14 can also trigger that, as you can see here, degrade CIP1, and therefore activate a NIC activity by stabilizing the protein. Okay? So we believe that's really the original function of these proteins, and cell death is probably secondary role. And why this is important, because if you treat cells with IP antagonist and you degrade CIP, you upregulate NIC, you're going to lead to upregulation of inflammatory cytokines. And that can be used in cancer immunity, because what you do there, you can combine with checkpoint inhibitors, PD-L1, and here's anti-PD-1, PD-L1 antibody, moderate activity, or you can use IP antagonist here, LCL-161. Again, moderate activity. If you combine them, you have much better effect, survival of this multiple myelomas in mice with multiple myelomas. So because what you do here, you inhibit checkpoint, and then you activate non-canonical FKB by IP antagonist, and then immune activation will then invoke this potent anti-tumor phagocytic activity. So this is actually right now IP antagonists are not considered for this direct caspase activation, but more for this process of immune activation and creating more inflammatory environment for tumor where other compounds could act like PD-L1 or PD-1 antibody. Again, one more application that we are currently looking is the HIV. Again, HIV treatment is now getting more and more efficacious, right? And people survive longer with HIV because there's protease and other cocktail of inhibitors. However, the HIV goes into latency. And it was found that NF-kappa B, non-canonical NF-kappa B, can wake up HIV latency and produce active viral particles. And there you go with the proper antiviral treatment and completely eliminate the virus. And so now we are collaborating with the international group to see if our IP antagonists can be used to activate non-canonical FKB signaling. We're doing experiments with the rhesus macaques, and therefore, if that shows fruitful, then we can start a clinical trial with HIV patients to eliminate them of the dormant HIV forever. Okay? So just shows you how we started with one way of looking at a reagent or compound, and then biology takes us to totally different direction. And so as long as we can help patients, we are happy. Or although the initial idea was to really work on the cancer, but unfortunately, unlike BCL2, there is no signature expression profile for IP proteins. So there is no easy way to detect which tumors to test and to target. The same thing with the death receptor agonist. Trail receptor 1 and 2, or even decoy receptors. Now we know that the profile of expression is not such that would indicate these are the tumors we could treat. The same with IPs that are present in most of the tumors. And that makes it more difficult. And I think combining that for tumor immunity and promoting tumor inflammation, it's a better way of doing that. And that's why we're using SMAC-medics mostly in a tumor-promoting inflammation sense to see if that's going to then help to get rid of the tumors. Okay. So I will stop here again for a second to drink a sip of water, and then invite any questions for this part, if there are any, or continue.
Okay, guys, any questions? You can start placing your questions anyway in the chat, just for us to register. Of course, I will ask you to open the mic and camera and ask Domagoj at the end, but feel free to, to write your question in the chat. Domagoj, I have a quick question based on this pro-inflammatory property of the IEPs in cancer. So some cancers benefit from inflammation. Others, of course, has a detrimental effect. How do you sort this out?
Yeah, I see what you mean.
Is it like a test and run thing, or?
I think there are some tumors that are called immune desert, that there is really very little of immunity there. There are almost no T cells. There is no macrophages, no inflammation. And they grow and proliferate. The idea is to go to those settings when
Okay.
there is no inflammation and then treat those tumors with the reagents that would promote inflammation and this kind of tumor inflammatory environment, and that way help get rid of the tumors. So
All
we
right
would
then.
not go to the others. I don't know if this would be good like for multiple myelomas or something, but so definitely there are types of breast cancer and others that would benefit from this.
No, I mean, for those, I think it's more obvious because, yeah, as you said, there's no much infiltrating, inflammatory immunity cells. But even for some that is using inflammation to angiogenesis or things like that, do you believe you might be changing the inflammatory profile? And then even then you may help.
Sure, it is possible. It is possible. I think it's been very difficult, as you know, after PD-1, PD-L1, there is no real drug, approved in this space. A lot of combinations have been tried, including agonism of TNF family members and TNF receptor family members, right?
Right.
That for the similar thing, activate non-canonical NF-κB signaling and promote inflammatory milieu in this case. I don't think it's clear, at least it's not to me, you know, when do you stop with that and when it's detrimental. Right now, in a lot of tumors, if you deem them to be immune silent, most likely they will benefit by promoting this inflammatory environment.
Great, great. Thank you very much.
Okay, so now we're going to switch to inflammation. Okay, so the way we think about inflammation, many ways. One way is you think that there is infection or tissue injury that will lead to low-grade inflammatory response. Okay, and so now what happens is you have pathogen clearance with various anti-pathogen mechanisms or tissue repair, and then you restore it to normal tissue and homeostasis is restored. However, if you have persistent stimuli, strong stimuli or prolonged, right, then you can incur tissue damage marked by cell death. You have release of DAMPs, these dangerous sociomolecular patterns, and cytokines. And then you have amplification inflammatory response through the macrophage and lymphocyte recruitment. Okay? And we think that this is often the case in processes including inflammatory bowel disease or psoriasis and other diseases. And definitely therapeutic intervention could be beneficial there. So interestingly, when we talk about inflammation, we are introducing other forms of cell death, not just apoptosis. And the one I want to introduce today is necroptosis. And while apoptosis is clear, dying cells engulfed by neighbors, there is limited inflammation, and core pathway components are well defined, caspases, BCL2IP proteins, and we have already drugs in this case. For necrosis is less known. It can be physical, ischemic, or inflammatory, and inflammatory necrosis can be either pyroptosis, mediated by caspases 1 and 11, gasdermin D, or it can be necroptosis, mediated by kinases RIP1 and RIP3, when caspases are inhibited. There are known triggers such as tumor necrosis factor, toll-like receptors, by many others as well. So I will introduce now a pathway again, but look at it more from the necroptosis perspective. Okay. In this case, again, we have TNF or related ligand binding to the receptors, TNFR1 binds TRAD, TRAF2, and then CIP1 will then bind to RIP1, ubiquitinated. That will allow NEMO recruitment and activation of NF-κB MAP kinases. However, there are factors such as deubiquitinase A20 that can block that process and prevent this. And so then if NF-κB is inhibited or over time, RIP1, receptor interacting protein 1, can move to the cytoplasm, and then it binds different set of adapters, FAD, and then caspase 8, and that will lead to activation of caspase 8, which then, as we know, can activate cleave caspases 3 and 7, and lead to apoptosis. However, if caspase 8 is inhibited or insufficiently activated, then RIP1 can move and then can auto-activate by adopting active conformation with autophosphorylate itself, as serine 166. Okay, and so now phosphorylate RIP3, RIP1 can bind and recruit RIP3 that can also autophosphorylate, and then it can bind and phosphorylate MLKL, which is pseudokinase, and then that goes to the plasma membranes and leads to necroptosis. Okay, and this is induced can be by TNF family members, but also by toll-like receptors like TLR4 and others. In this case, adaptor retrieve that can directly bind RIP3 or RIP1. But there are other factors like ZBP1. I will not talk about that one today. And then tissue damage, which is induced by either stroke, heart attack, and various different ischemic injury can also lead to activation of apoptosis and necroptosis. In necroptosis case leading to rapid membrane permeabilization, release of DAMPs, such as Al1 alpha, HMGB1, and so forth. And we know that this is very dynamic process, and RIP1 can be both active in apoptosis and necroptosis in this case. Now, we were thinking, if we want to prevent this tissue damage, for example, what would be the right target for this approach? Would it be RIP1 or RIP3? Since RIP1 is broadly present in other pathways, although this is not kinase dependent, this is just a scaffolding function, kinase activity is here. First, people thought RIP3 is a better target for drug discovery. However, what Kim Newton, our colleague Genentech, and Vishwa Dixit found that actually you cannot target kinase activity of RIP3, because when they made kinase dead version of RIP3, that caused embryonic lethality. Even if they inactivate RIP3 kinase activity in adults, it was also toxic. And then GSK developed RIP3 inhibitors, also toxic. So it seems that if you inactivate RIP3 kinase activity, then you activate RIP1 dependent caspase activation and apoptosis. And that's why you have this lethality. And true, if you delete caspase 8 together with kinase that you can rescue this process. So it's really caspase dependent in this matter. However, on the other hand, RIP1 kinase that is fine. There is no problem with inactivating RIP1 kinase activity. And here is a paradox. If you have RIP1 knockout, full body knockout, is dead, perinatal lethal. They die a few days after death. But RIP3 knockout are viable, no problem. They live normal life. On the other hand, if you inactivate RIP3 kinase activity and make this RIP3 kinase dead knocking, they're dead. But RIP1 kinase dead knocking are completely viable, and there's no problem with that. As you can see here, RIP1 cells, these are macrophages for RIP1 kinase dead mice or RIP3 knockouts, efficiently block necroptosis in this case. And so based on that, we decided RIP1 is a better target and not RIP3 because it causes toxicity. And we focus on targeting RIP1. First, we wanted to validate that it would make sense by doing simple experiment, which is a TNF sears. So systemic inflammatory response syndrome. So if you give animals a large amount of TNF, they will die within 10 to 12 hours from that. However, if you give the same TNF amount to RIP1 kinase dead mice, they largely survive, no problem. Same thing with RIP3, okay? The wild type mice die, but RIP3 knockouts survive. So that really validated that these factors really matter for TNF biology in vivo. And we use these models for our in vivo investigation in disease settings to see where this process plays a role and makes sense to target. Okay? And so mostly we're going to talk about targeting RIP1. But this is by far not the only kinase that is relevant in inflammation. Here, this is the review we published a couple of years ago to show you list all these different kinases that have been or could be targeted in inflammatory diseases. There is a lot of them. For example, JAK inhibitors are the best example. There are approved drugs targeting JAK1 only or JAK1, JAK2 pan-JAK inhibitors, as well as for TYK inhibitors. There is efforts to target IRAK1 and IRAK4. There is efforts to target TBK1, NIC, a lot of them, TPL2. And then there is also efforts to write RIP1 and RIP3. So I will focus on RIP1 kinase because that's the only kinase that plays a role in cell death by its kinase activity and it can be targeted for the benefit of the patients. In this other case, these kinases actually promote survival and proliferation. And when you target them, then you cause cell death that in some cases actually can be rescued by RIP1 inhibitors. Okay? So we develop a series of agents that can target RIP1. And I'm showing you here one compound that we called GNE684 that we developed, especially that we can use it in mouse models of diseases. And you can see that it binds and blocks both human and mouse RIP1, nice activity, and also has nice pharmacological properties, so it can be used in this setting. And then we tested it in a TNF hypothermia. In this case, we made a slightly modified version to speed it up even more. So not just putting TNF, but also put caspase inhibitor ZVAT. So ZVAT inhibits caspases. And then you promote strong necroptosis. In this case, by 4 hours, all animals are dead. And in this experiment, even after 2 hours, they lost over 10 degrees of body temperature. And then we give them various amounts of RIP1 inhibitor. And we see even at 1 mg per kg, we see almost 40% inhibition, but everything about 5 mg per kg, almost complete inhibition of this hypothermia and animals okay and saved and survived. What is important to note that RIP1 kinase activity does not matter for NF-κB activation, and therefore it should be safe. So you see the wild type macrophages or RIP1 kinase dead macrophages, treat with TNF for various times. RIP1 levels very comparable, other wild type or kinase dead, and you see phospho kappa B, total kappa B, JNK, ERK, all that activated in very comparable fashion, regardless if animals are wild type or RIP1 kinase dead. Okay. Next, we also look at the different lymphocyte subsets from these mice, and whether they come from wild type or RIP1 kinase dead mice. You see all these different from lymph nodes or spleen, the B cells, T cells, all the same, no problem. Okay, and then also we tested our compound in the context of TNF. As you can see here, that there is no problem with any of these factors. CIP2 is upregulated, A20 upregulated, and even if we use 200 micromolar of the compound, still no toxic effect, no effect on NF-κB induced activation of these processes. Okay, so that tells us that we can have safe reagents that can be used for treatment of various diseases. But the question was, which disease you want to treat, okay? And so now over the last 10, 15 years, number of investigators throughout the world have tested that hypothesis and tested various diseases. And we, believe it or not, tested them in all these models except for the Alzheimer's disease. In all the others, we have tested RIP1 inhibitors. And so the results are not what we expected to be. For example, I'll show you a few examples where it was claimed that these compounds play a role, but we show it doesn't, like pancreatitis or cancer. For example, it was reported that RIP3 knockouts are somewhat protected in this pancreatitis model, which is induced by treated mice with serolin. Okay. However, we show that RIP3 knockouts are no different from the wild types, PBS or serolin treatment, no difference, and also RIP1 kinase dead mice, also no difference. So it really tells you that in this kind of, when we look at the serum amylase activity, there is no levels and no difference. Therefore, doesn't play a role here. The other example was cancer, because of course everyone wants to solve the cancer, and the idea was that necroptosis would promote pancreatic oncogenesis, and that RIP1 kinase drives macrophage-mediated adaptive immune tolerance, and there is also necroptosis linked to metastasis and all that. And then RIP1 inhibitors can be combined with PD-1 antibody to allow prolonged survival of the mice in this model. Okay, and so based on the GSK designed another compound special cancer study and wanted to start the treatment. Okay, we were surprised, but so when you look at the apoptotic necroptotic pathway here, right? You know, we have various way of targeting this with cell death related treatments. As I said, for death receptors, we have these antibodies that would agonize and trigger cell death. For IPs, we have IP antagonists that should activate caspase A or other caspases. Nutlin's type of reagents, they would activate p53 and break inhibition for MDM2. And then BCL2 antagonist, we have venetoclax as the only approved drug. Some are still being explored. However, the question is whether necroptosis here, pathway regulated by RIP1 and RIP3, can also play a role. And so we want to explore that, because if we have these reagents, we should be able to use them in a cancer setting. So we have done this in the same fashion as the other authors, and use the most physiological way of testing cancer, which is in GEMS, or genetically engineered mouse models of cancer. So these are spontaneously cancer that mouse develop when they have KRAS and other mutations, P16, P19, or KRAS and P53 mutation, okay? And so these animals, after 3 or 4 months, develop tumors. However, if we treat them with RIP1 inhibitor, we saw no difference in survival, and we saw no difference in the tumor growth. And we actually did a couple of models and couple of compounds, including NEC1A, no effect in any of these studies. Okay, so I'm just showing you a summary here because we did much more. We also followed on this report that RIP1 kinase that can modify macrophage signaling and lead to activation of STAT1. However, we didn't see anything. There is no different expression profile from the wild type of RIP1 kinase dead knock-in mice for all these factors that were reported, and there was no STAT1 phosphorylation by themselves. And we used our compounds, or GSK compounds, in both of these cases. They're very close in their affinity for mouse RIP1 and efficacy, but there is no effect here. So basically, we concluded there is no point in pursuing that, although we had to spend a couple of years on doing these experiments. But we did it to convince ourselves this is not the right disease to go for, right? And then actually, GSK didn't even treat patients, halted enrollment, and they blocked this because I think they realized, as us, that there is no potential here. Yeah? And so at least patients not given something that would not help them. So at least that much. Okay. So as we go for this list of diseases, I said GSK tried here. We also show lung mets are not the real thing. But what we did show that it does matter in acute indications, in kidney ischemia, in myocardial infarction. That's where it really matters, okay? And in stroke, and I'll share some of the data. But in psoriasis, on pancreatitis, it really does not, okay? And so this was also published here, so you can go back to these papers if you want to read more. We also published that it doesn't matter for most viral clearances or mere reproduction degeneration, and those are these references. However, ALS was not known at the time. Now it's known that it doesn't work, unfortunately. And COVID trial Sanofi did, I should update this, actually it's known now that the RIP1 inhibitor did not work for COVID, but it was very close. They actually show number of biomarkers showed a reduction of inflammatory factors were greatly reduced by RIP1 inhibition, but they didn't meet the clinical endpoints regarding hospitalizations and the lung function and so forth. Okay. Now, the one we wanted to explore is also ALS, because that was also published as being really the one way to go. However, we did SOD1 transgenic model, which is a classical model for ALS. As you can see here, when we did it across SOD1 transgenic mice with RIP1 kinase dead, there is no significant difference for any neurological parameters we measured, myelin areas, or any other. Here is spinal cord inflammation. All was similar, and we reported that also because we wanted the wider public to know that we don't believe in this. And sure enough, just recently, it was revealed that a Denali trial of ALS did not meet its primary endpoint. However, there are other diseases where this matters, and you have to go where there is a tissue damage, okay? And so to do that, we made another strain, and we made the rats that are kinase dead. So we made RIP1 kinase dead rats, okay? And in this case, again, you see very comparable levels of various B cells, T cells, lymphocytes, spleen, lymph node, just in wild types or RIP1 kinase dead mice. Also, there is no problem with the activation of NF-kappa B. We took BMDMs from several wild type or kinase dead rats, no problem. However, they were protected in a TNF-induced series. And interestingly here, how different mice are from rats. Mice can tolerate up to 7 or 8 degrees of loss of body temperature. Rats, even after 2.5 degrees, 3, they start dying. You know? So it's much smaller, this range in rats. And I do not know why, but even after 2 or 3 degrees, they start feeling bad. And mice can tolerate it very well. Nevertheless, if TNF was given to RIP1 kinase dead rats, either males or females, they are fine. Either TNF only or TNF Ziva, they all survived. So that really kind of validated this model. So knowing that we have a model that is responsive to necroptosis and blocks it, but doesn't affect normal homeostasis, normal activation of kappa B, we went to the brain and also did a micro CT to investigate cerebrovascular anatomy of wild type or RIP1 kinase dead rats. And you can see here there is no problem. And then we did experiment where we subjected them to the stroke, which is this major occlusion model, MCAO. And then you can see here, days after that, in a wild type mice, you have this lesion that's greatly reduced in RIP1 kinase dead. So in this case, when you lower, it's better because you're reducing the lesion in the brain of these rats, okay? And then we can also look at the myelin stain in a wild type rats or in RIP1 kinase dead. And you can see even from this picture, it's clearly preserved in RIP1 kinase dead, and that's a graphed and tabulated, quantified here. So it shows you that when you have ischemic damage, like a kidney damage, heart attack, or stroke, this is where RIP1 kinase inhibitors could be effective and help patients. It's just trick how to dose it at the right time in the right place. Okay. So to summarize this part, we have shown previously effect in the kidney injury and heart injury, that there is nothing in cancer or viral clearance, and also there is beneficial effect in stroke, but not in ALS. Okay, and so I want to now turn our attention to inflammatory diseases. Okay, so and chronic indications such as skin inflammation and IBD, rheumatoid arthritis, maybe in some other time. As I said, to summarize, we show no effect on testicular degeneration or viral infections, no effect on tumor growth metastasis. Okay, so to study this, we turn to genetics. We turn to mutations that occur spontaneously in humans And I can tell you, NF-kappa B pathway is heavily mutated in people with inflammatory bowel diseases and skin inflammatory diseases. So we want to mimic the mutations that occur in human population and make mouse models that mimic those mutations, and then therefore have that physiological translatability and test our reagents in that setting. And sometimes it is in sometimes it's human, and sometimes we also look at the mouse mutations. Here, the way we demonstrate activity here is by looking at phosphoryp1 staining, and we see some moderate staining in psoriasis, not great. And then we use the sharpen model, which is mice that spontaneously develop a skin inflammation, okay? And so then the first pilot experiment was just take these mice when they are 6 weeks old, they already have skin inflammation, and treat them with a compound for just 4 days. As you can see, even in 4 days, we see nice reduction in skin inflammation in total skin, and especially in ventral cervical skin, okay? So we said, well, if we do it only for 4 days, let's do a longer treatment and see what will be effect in that case, okay? And so now we did treatment for 4 weeks, okay? Again, this was done after mice already developed skin inflammation, okay? And in this case, you see that 4 weeks or no, 26 days of treatment with G684 really reduces this inflammation. Histology looks just like wild type mice, and then here is sharpen mutant CPDM mice, very strong skin inflammation, okay? And that's quantitative here. As you can see, this mouse have a lot of inflammation on the chest area. Those with vehicle, however, if it's those with our compound RIP1 inhibitor, almost nothing there. So then we want to know what processes are involved in this skin inflammation. And first we look at the caspase activation. And we see there is caspase activation here, actually. So in this case, you have a strong caspase 3 cleavage, quantitated here. And then we look at RIP3 phosphorylation. So you have also phospho-RIP3. So remarkably, in this setting, you have both apoptosis and necroptosis occurring in the same mouse, in the same skin, not maybe in the same cell, we didn't see in the same cell, but the mice experience both apoptosis and necroptosis at the same time. When sharpen is mutated, that means you don't have proper linear ubiquitination and you don't have proper NF-kappa B activation, okay? Now, interestingly, that sharpen deficient mice have other diseases, not just skin inflammation. And one of them is very serious condition called eosinophilic esophagitis, okay? Eosinophilic esophagitis is characterized by the infiltration of eosinophils in esophagus, as you can see, increased number in CPDM, in the mutant mice, and then thickening of their esophagus, okay? So we want to see whether, RIP1 kinase activity plays a role there. So we treat them, with the same, in the same mice, we look, and we see some reduction of inflammation in esophagus, but not statistically significant. So some partial reduction in that case, nothing like skin. Again, in the same mouse, okay? But then we turn to the other organs and look at the liver. In liver, we see kind of halfway, okay? You see a reduction of this process, better than in esophagus, but not as good as in skin. And so we were thinking, well, why is that, okay? And then we stained liver sections for caspase 3. And again, we see clip caspase 3 here, that's reduced by compound. So it tells you more cell death, then RIP1 has more role, okay? And so then in skin, there's a lot of cell death, a lot of clip caspase 3 and a RIP3 activation, and blocking RIP1, very efficacious there. In liver, there is some cell death, and at least partially, this disease relies liver inflammation on activation of caspases and RIP1 activity, so therefore you can reduce that. But in esophagus, it seems that RIP1 does not play a role, there is no cell death, and therefore you don't expect to see a rescue in that setting. So this is a nice example, I think, that you see multiple cell death pathways and multiple organs in the same animal that from one mutation affected in different way. So just tells us how complex cell death pathways can be in disease settings and affect different organs differently and utilize different pathways. Okay. The other model that we use is a NEMO model developed by Manolis Pasparakis originally. So in this case, you can delete NEMO, which is NF-κB essential modifier, in intestinal epithelial cells. You delete it in intestinal epithelial cells, you will block NF-κB activation, and then you activate RIP1 and inflammatory cell death. As you can see here, if we have a NEMO wild type and RIP1 wild type, everything is fine. However, if you have NEMO conditional knockout, so you delete it in intestinal epithelial cells, then you see massive destruction in colon, and you see some ileitis, mostly you see loss of Paneth cells in the ileum. However, you block RIP1 by crossing mice to RIP1 kinase dead knocking, and you restore that completely. Colon looks fine, Paneth cells are back, and that can be quantified here. So it's really remarkable how that it can be restored in that case. And then we look at the inflammatory factors here. We can see a lot of them upregulated when you don't have NEMO, and it's all really cell death driven. These are the in red, especially in the GCSF, CCL2, CXCL1. However, if you have NEMO knockout and RIP1 kinase dead, then goes back to the black lines to the wild type form. So RIP1 really restores that. And we see that in this case, we can see RIP1 phosphorylation in this case. This is ileum after several days. From these mice, you see nice RIP1 phosphorylation here, and of course, you cross to RIP1 kinase dead, there is no RIP1 phosphorylation. Okay, so that really demonstrates this is on target and how you reduce RIP1 activity, you reduce cell death, and you reduce tissue inflammation in this case. And here we show you that actually we can do that both with the compounds or with the kinase dead. So you see here in this one, that's a NEMO conditional knockout. So you see inflammation, colitis, and ileitis. However, if you treat with compound RIP1 inhibitor, reduce that both in colon and ileum, or if you cross NEMO conditional knockouts to RIP1 kinase dead, you reduce that. Basically the same level of protection from the compound or genetic model. And then you can see here when you see this damage histology and completely restored. And again, we looked here for various factors, and we see mostly apoptosis in this case. So it's a massive caspase activation in this case that's almost completely blocked and obliterated by genetic or chemical inhibition of RIP1. Okay. So in this case, it seems, at least in the gut, it's mostly apoptosis that plays a role. We looked at additional factors that play a role, especially when they're linked to human disease. ATG16L1 is one example. That's the autophagy regulator. That's the most frequently mutated gene in Crohn's disease. And actually, especially people of European descent, almost 35 to 40% of them that have this mutation have more severe form of Crohn's disease. And this mutation is T302A, so 302-alanine, and makes this protein good substrate for caspases. And then caspases cleave it, and then you have deregulated autophagy, and then you have Paneth cells messed up here. You see these are Paneth cells in wild type, and there are Paneth cells here. And Paneth cells are critical for detection of microbiome and for regulation. When you don't have it, patients are much worse off. And again, so we made mice that have ATG16L1 deletion in intestinal epithelial cells, and they're very sensitive to TNF. See, just TNF, nothing else. In 4 hours, they all die. However, if you get them compound, very nice rescue. So that's from these open symbols to closed symbols. Another example I want to give you is with A20. It's a deubiquitinase that regulates TNF signaling, also mutated. This is a young woman with a also not specified IBD, I think likely Crohn's disease. And in this case, again, more severe phenotype. When we, again, can take A20 out by doing intestinal epithelial knockout, very sensitive to TNF and very nicely rescued by a RIP1 inhibition. And overall, when we look at this pathway, we see there is a variety of proteins that are regulated TNF, RIP1 signaling pathway. So many of them are mutated, and there are mutations people have HOIP and HOIL1 mutations, autolin mutations, XIP mutations now linked to the early onset IBD. If you have cleavage-resistant RIP1, that's linked to autoinflammation, NEMO hypomorphic mutations, ATE1 mutations, and so forth. So it really tells us that, and so now our idea is that we should sequence all these patients and really identify who's most likely to benefit from this treatment, because we can see who has mutations that would activate their cell death in RIP1 dependent fashion. Okay. So in addition to this, I want to cover in the last segment a disease that maybe doesn't have so strong genetic component, and we haven't studied it yet before, but I think it's a really remarkable disease and there is great unmet medical need there. And that is acute graft versus host disease. Okay, there is definitely unmet medical need. That happens when a donor-mediated immune destruction of host tissue, when people either, cancer treatment, radiation, or some other receive transplant of hematopoietic stem cells, and hopefully it helps them, but sometimes it doesn't, and then you have graft versus host disease, and it's very, very bad. It mostly affects intestines, but also skin and liver. There is poor response from patients, mostly steroids, although right now JAK1 inhibitors have started being used just last year. And these immunosuppressants like steroids also cause adverse events and toxicities. So definitely there is a need for a non-immunosuppressive drug. And RIP1 is, because, as you saw, doesn't affect viral infection, doesn't affect NF-κB activation, has no immunosuppressive properties. And this is the, again, picture of the early acute GVHD intestine, and this advanced, very painful, very bad. Okay, so to investigate whether RIP1 pathway is active here, we again wanted to see human patients and investigate RIP1 activation in that. So we used phospho-RIP1 antibody to stain samples from a variety of GVHD patients. GVHD patients are ranked by Lerner score. Lerner score indicates histopathological severity. So higher the score, higher severity. In this case, you see remarkably that higher RIP1, phospho-RIP1 score, higher Lerner score. So there is really nice linear correlation of RIP1 activation and histopathological severity of disease. Okay, and I'm giving you examples what RIP1 strong staining is and RIP1 no staining. So we have all variety here, so that this really is a really severe disease, and this is mild or almost no disease. Even more remarkably, this correlates with non-relapse mortality. So in this case, when you have higher non-relapse mortality, you have more phospho-RIP1 in the gut of these patients. And then when you have lower non-relapse mortality, patients doing better, they have lower score for phospho-RIP1. So that really tells you directly from the patient that we can see how important the pathway is. So I will skip all the cellular studies here, and I'll go straight into in vivo work, because I think it is pretty remarkable and shows what can be done here. So these models of GVHD rely on HLH mismatch, okay? And these are, of all the models, the most relevant and clinically translatable models, because really the most closely mimics what's happening in the patients. So what we do here, we irradiate mice, what we call day -1, and then we give them bone marrow transplant, and then after that we give them compound for 2 weeks, and then we follow them for another 2 weeks with nothing, and then we end the study and we monitor survival. We monitor other factors, but in this case was survival. As you can see here, if you have a major mismatch or minor mismatch in this case, if you give them syngeneic transplant of bone marrow and splenocytes, there is no GVHD, so there is no problem. But you give them allogeneic, then you develop GVHD, and if you treat them with vehicle, all die within a month, all mice die. However, if you give them compound for just first 2 weeks, majority of them survive. And this is in a major mismatch model. In a minor mismatch model, again, 60% of the animals will die after 4 to 5 weeks. None of them die if they got RIP1 inhibitor. Okay, so this is more therapeutic setting, I mean preventative setting, and we have done this also, of course, with RIP1 kinase dead mice and so forth. But we want to do that in a more therapeutic setting. Okay, so in this case, we would start earlier. So let's say irradiation day -8, and then after that, bone marrow transplant, and then for a week, no treatment with compound. So they develop disease in this case. Then you give them compound for 2 weeks, and then you follow. So much longer study. So what we start here is from the day of compound treatment. And you see they start dying right away because they already had disease developing for over a week. Whereas here, it takes them a week. But in this, we indicate we start right here. And they start dying if they have allogeneic transplant and receive vehicle. But if they receive compound, there is a rescue again, and significant rescue. So that means in the therapeutic setting, we can also use that paradigm and treat mice and hopefully treat patients in the same fashion. Okay. And so I want to end with a little bit of a mechanistic studies that we did here. We just published that in end of the last year. So you can see more details, much more immunology there. But we treat organoids derived from these patients, and we see phospho-RIP1, we see cell death, and that's nicely rescued here by RIP1 inhibitor, reduces this phospho-RIP1. We see also cleave caspase 8 that can be nicely reduced by RIP1 inhibitor. And what we use here is combination of TNF alpha interferon gamma, because I believe that's the most relevant treatment, because TNF alpha interferon gamma are both elevated in the intestines of patients with GVHD. Okay, but what's interesting here is you see cell death in this case and reduction of viability. Now you give them RIP1 inhibitor, you restore viability. You give them imricasan, caspase inhibitor, you restore viability. You combine it, there is no added benefit. Okay, so whatever caspase inhibition or RIP1 inhibition provides the same benefit in this case. And true enough, what we see here is in syngeneic transplant, there is no activation of cell death, there is no tissue damage. In allogeneic, 9 days post transplant, we see cleave caspase 8, we see cleave caspase 3. We do not see necroptosis in this case. There is no phospho-RIP3, there is no phospho-MLKL. This is all apoptosis. And sure enough, the same samples that I showed you for phospho-RIP1, now we did cleave caspase 3, and again, we see nice activation of cleave caspases in this case in the same patients, just correlating and mimicking very nicely phospho-RIP1. So in this disease, we thought it's going to be mixed, but it's all, especially intestines, it's all apoptosis. There is no necroptosis, unlike what we see sometimes in skin. We see necroptosis in kidney damage, but in this case, intestinal damage, either being pneumocolitis or with GVHD, it's almost all exclusively apoptosis. Okay, and so I hope I showed you for this part of the presentation, gut inflammation and damage really to RIP1 activation, and then activates both pathways, predominantly caspase 8 here, and we believe that's really clear in Crohn's disease, in GVHD, and that RIP1 kinase inhibitors can be beneficial here. And we see that it's not just there, but skin inflammation, where we see mixed of both necroptosis and apoptosis, again, causing this inflammation, inflammatory cell death, and tissue damage, as is seen in various diseases, lichen planus, Stevens-Johnson syndrome, toxic epidermal necrolysis, but not psoriasis. Not psoriasis, because psoriasis, there is almost no cell death and very little RIP1 activation. And so we are guided by this principle. If there is a tissue damage and cell death, that means these factors should work there, and that's guiding us in our clinical approach to treating patients. And so then, as I said, TNFR1, when it's bound by TNF, and then how do we see this necroptosis? IP antagonist and ZVAD all come to prevent RIP1 would be ubiquitinate. But then if you have IP antagonist in ZWAT, that does not happen. RIP1 is free to go to the cytoplasm and then binds caspase 8 with FAD and RIP, which leads to this activation. And however, then it can bind and activate, auto-activate RIP3 and MLKL and lead to necroptosis in this case. And we hope that a RIP1 inhibitor such as GNE684 can break that and both block apoptosis on this end and necroptosis on the other end, and therefore help patients suffering from inflammatory diseases. At the end, I would like to thank a number of colleagues that contributed to these studies, especially Eugene and Tanya in my lab, and various others who led BCL2 and IP program. This was done also in collaboration with Vishwa Dixit and Kim Newton and Joshua as our pathologists at Nahel, who made the RIP1 inhibitors and Kurt IP antagonist, and Manolis for the gift of the Nemo conditional knockout mice. BCL2 project was done in collaboration with the Walter and Eliza Hall Institute and AbbVie. And I thank you for your attention and be ready to take any questions now.
Oh man, that was wonderful. Really cool. I have few questions myself. Ricardo already raised his hand. We have many questions in the chat, including for a couple of students that mentioned they could not open the mic for some reason. So let's start with Ricardo, then maybe we read those questions from the chat and then move on to anybody else that can raise hands, okay? All right. Okay, go ahead, Ricardo. Maybe if you
stop sharing your presentation, it's better to see.
Yeah, you can see the students. There is this, we have this student here, Domagoj, like a bird old man that's been following here.
Oh, look at this. Peter is there. I saw Peter a couple of weeks ago. So
you were lucky,
by now, Peter can give my talk by now. He heard it so many times, he can give my talk. Right, Peter?
Okay, so go ahead, Ricardo.
Thanks, Domagoj, for the amazing lecture. Very hopeful that this works in humans and you can treat many diseases. So just a mechanistic question. So how blocking the kinase blocks caspase 8 activation? So I remember that RIPK1 autophosphorylates and helps phosphorylating RIPK3, or at least recruiting RIPK3 to the complex. I don't remember well how the kinase is important for caspase activation. I thought it was independent.
Well, caspase 8 can be independent and can be RIP1 dependent. There are two forms, because if it goes through TRAD, which is another adapter downstream of TNF, then it would be RIP1 independent, and then you can activate caspase 8 directly that way. But if you involve RIP1, then RIP1, it'll be part of that complex, and its kinase activity is really important to maintain this complex where FAD is also and caspase is active. And it seems that you block RIP1 kinase activity, you disrupt this complex, and therefore you diminish caspase activity, not by directly inhibiting, but by dissolving the complex where caspase 8 is active. So, I mean, it's more stoichiometrically, it's no direct inhibition, but FAD, RIP1, and kinase, caspase 8 need to be in this complex together, and when you disrupt that, then a caspase 8 is not properly active, and then you don't have downstream apoptosis.
Which is different from taking RIPK1 out of the question there in the complex, right? So do you know what's the target of the RIPK1 phosphorylation there to keep itself?
It really it's been studied a long time, and it's clear now it's only itself, and that's how it regulates this activity. It can be phosphorylated by other kinases, for example, TBK1, TAC1, MK2 can phosphorylate RIP1, but that's in a complex one, and that phosphorylation actually inhibits cell death and is meant to keep RIP1 in the complex. But phosphorylation at 166 serine, that's the one that allows activity of the cell death promoting complex, and that's where its own activity is required.
Thanks.
Okay, so I'm gonna start reading some of the questions from bottom to the top, but I'm not gonna read any questions for the students that are already here and can open the mic. Okay, so you start raising your hands and ask the question yourselves. Okay, so but I'm gonna do that for Maniz. Maniz said that thank you very much, wonderful lecture. What are the strategies to overcome the challenge for solid tumors as gliomas, for instance, not just the BBB, of course, about clinical studies in human? There are a lot of phase 3. And what about those regimens? So you can figure that out.
Yeah, I think there are efforts to target solid tumors, absolutely. But to be honest with you, they don't rely at the moment so much on cell death or on activation of cell death directly. Okay. As far as I know from my company and many other companies, that it's mostly KRAS and some other proliferative pathways that are employed here as directly blocking those oncogenes or Hippo pathway. On the other hand, it's promoting tumor immunity, and by activating NF-kappa B in a way that would stimulate release of pro-inflammatory cytokines and create more inflammatory environment for tumors where the checkpoint inhibitors could work. BCLX is probably the best hope to do cancer killing cancer cells, but unfortunately that toxicity is there. MCL1 is the other one. Again, there are toxicities, but I know companies have stopped straightforward inhibitors, but there are efforts to do degraders of MCL1 that would maybe not completely take it out, but lower the level, and therefore maybe sensitize them to other treatments.
Domagoj, but you mentioned in your talk strategies to inhibit RIP1 or 3 in cancer. But what about like stimulating the necroptosis?
Yeah, that's also been published. I think honestly that would be dangerous because when I see necroptosis activation, that's associated with tissue damage in heart and kidneys. And so I don't see how would you develop this reagent that would just go to cancer cell and not touch other organs if it's going to be systemically administered, right? If you can do something magically to keep it in the tumors, okay.
Yeah.
But if you're going to have systemic distribution of this, you're going to cause damage to macrophages, to possibly T cells, but definitely kidneys, heart, and so forth. So therefore, I'm skeptical about that.
Yeah, but you're not aware of people trying to do that in the solid tumor, like just hit a bit of the solid tumor, things like that.
I am not aware of any single company doing that. I know there are people, academic labs doing that, publishing. Okay. Absolutely, you can publish on that, but I have zero translatability to cancer treatment. Okay.
Okay. All right. That's good to know because we are most used to read academic papers and then we think, oh, okay, that's easy now. But that's why we invite you every single course because you know how to say, this is not, yeah.
Believe me, maybe it's a little depressing, but you know what is AACR, right? Major American Cancer Research.
Yes.
There was almost no cell death there,
Oh.
Almost no cell death. It used to be 2 or 3 sessions dedicated just to cell death. Now, very little, few posters here and there, just tells you how the field, cancer field, moved towards, targeting oncogenes, degrading some factors, and tumor immunity. That's it. And I think for cell death, inflammatory diseases, autoimmunity, tissue damage, death, that's where the focus should be, my opinion.
All right, that's important to know. And here comes Doug. Hey, Doug, how's it going? Can you hear it? I think he's getting in the room.
Yeah.
Okay, let's go, Leticia.
Hi, great class, Professor. My question is similar to Monique's question, because you comment that the increase in expression of BCL2 are in hematological tumors, but I saw that breast, gastric, and prostate cancer, we also upregulate of BCL2. But apparently, if you use the BCL2 blocker, they seem to respond better in hematological tumors than solid ones. And we know that solid tumors can promote the microenvironment that difficult the interaction with the drug. But in your opinion, what other explanation would have for the situation that the treatment seems show more greater efficacy against hematological tumors than solid?
I think for BCL2, the critical part is the expression levels of all these different proteins, So in hematological tumors, you don't have so much of MCL1 or BCLXL. They rely on BCL2 primarily for their survival. However, you go to solid tumors like a lung cancer, there is a lot of BCLXL expressed there. There is MCL1 also not so much. And that's the major reason why this approach doesn't work with venetoclax in solid tumors, because there are other factors that these tumors use for survival, not BCL2. That's the simplest explanation. So you should always, if you investigate those samples, either from mouse models, cell lines, or human, you should look at the whole panel of these BCL2 family members, pro and anti-apoptotic, and look at the balance. And I think it's going to be clear where venetoclax works, that's where BCL2 is the dominant factor, and when it doesn't, it's not. There's other factors, BCL, and MCL1.
Perfect, thank you.
Just 1 second, people. Doug is trying to open the mic and the camera, but is not being allowed for some reason. Márcio, você pode ajudar aqui a tentar resolver aqui? Será que ele precisa sair e entrar de novo? Está por aí, Márcio?
Eu tô. Quem o Doug?
Doug Green, é.
Ah, tá.
Se conseguir fazer aí daí. E põe ele já de co-host também, tá bom?
Tá bom. Só é que eu não sei se eu perdi alguma coisa, mas já acabou esse? Vai começar o outro?
Não, ainda tem mais umas perguntas. Nós vamos fazer uns 5 minutos break e depois começa.
Tá bom.
Right.
Eu vou ver, mas eu acho que não tem nada aqui, mas eu vou dar uma olhadinha.
Okay, Doug, Marcio is trying to help you. If possible, he's the host, whatever features here. But if not, I would ask you to exit and come back again. Okay, just a second.
I'd rather get questions from students than from Doug. Okay.
Yeah. Yeah. Okay, people. Maribel, I see another people. Caro, just turn the mic on, raise your hand, and ask your questions. Okay, please.
Thank you, Professor. Great presentation. My question is, can the efficacy of RIP1 inhibition as a single agent be used for autoimmune conditions such as rheumatoid arthritis, for example?
So we looked at rheumatoid arthritis, and we look at it 2 ways. First, we look at from human patients, that where there is how much of RIP1 activation we see there. We see some, but not great, okay? Second, we looked at the models of rheumatoid arthritis, and we some effect, 50% reduction, moderate effect, not great. Third, we look where anti-TNFs are approved, okay? And TNF blocking reagents. And they show some effect there, but not great. Based on all this, I think that RIP1 inhibition could provide some benefit, but would not be competitive because you have to see what else is available for the patients. And there are other things that are much better, JAK inhibitors, BTK inhibitors. There are other reagents, IL-23 blockers, IL-17 blockers. There are other reagents that show superior efficacy. In addition, in rheumatoid arthritis, there is not so much tissue damage and cell death and in some other diseases. So when we look at the totality of the data, we decided not to go for rheumatoid arthritis, but go to diseases where we see more tissue damage, more cell death, and activation of RIP1. Does that make sense to you?
Okay, thank you.
All right. Doug is keep trying to get into here. Let's see. Meanwhile, let me pose. Let me.
There's a person with your
Yeah,
hands raised.
Leticia. Okay. Yeah. Oh,
Caro.
Caro. Yeah, you go ahead, Caro, please.
Thank you for the good lecture. My question is like a cosmetic issue or concern on the wound healing on the skin, where we have some people suffering from keloid formation. Do you have any idea, or people doing research on that, like is keloid formation related to cell death? Because it might not be a big issue, but cosmetically it's affecting quite a number of people, because at times the keloids can be so huge you have some people maybe who have keloids after a burn, after having a severe burn, then the keloids become so big and they affect other parts of the body or some organs in the body.
It's a very good question. I have to admit we don't work on keloids, but I recognize the issue. My daughter has keloids, so I would really like to help her, but we don't have reagents for that. Regarding the burn, that's something we would consider, because definitely there is a tissue damage there, and I think very likely that there is a potential. We are looking, for example, in skin disease called pyoderma gangrenosum, which also has a painful skin ulcers, and we see RIP1 activation in those cases. However, it's not well developed regarding how to treat skin diseases unless they have strong inflammatory component, because when they have strong inflammatory component, then you can give them systematically as a pill or injectable antibody. However, if it's surface, then mostly topical treatments could be applied here. So I think it's a big opportunity, and it should be studied more, but I cannot tell you that we are doing that at the moment, and whether it's the pharmaceutical industry or cosmetics industry, someone should look into that, but I think it's not very well studied. Yeah.
Okay, I'm gonna write the Leticia Seraphin's question because she mentioned that she cannot open the microphone, but I saw Bruno and Lucas. You two be prepared to ask your own questions, okay? So she mentioned thanks for the class, and I think what she wants to know is a bit more about the possible side effects of body homeostasis when using any cell death target therapies. As you mentioned,
Yeah,
extensive.
it depends if you want to induce it or you want to inhibit it, right? When you want to induce it, like BCL2 antagonist, venetoclax, SMAC-mimetics, then yes, absolutely, you have to be worried about side effects. You know, for BCL2, luckily it's a manageable, moderate, and there is not much side effects. Is because there are other BCL2 family members that can take over the function. So, you know, mostly attacks the lymphomas, leukemias that rely on BCL2. Regarding SMAC-mimetics IP antagonist, there is concern because, and there is toxicity there when you use a high level, because you can then cause cytokine storm and activate NF-kappa B non-controllably. So it has to be dosed properly, you know, timing, amounts, and so forth. There is concern. Regarding RIP1 inhibitors, we do not have that concern because RIP1 kinase dead mice or rats are fine. There is no issue. So we don't see any target effect. But as with any compound, we do run preclinical trials, and anything before it goes into people, it goes for months and months into monkeys and rats and so forth to test for any unwanted effects. And there are compounds that have that, but it's more to do with the chemistry than the target, and those are not progressed. So only safe things go to people. I cannot hear you.
You are muted.
Okay, sorry. I said Lucas, Igor. You already left. So Igor has never
Hello.
shown his face. What about Lucas?
Thank you,
All right.
Professor. I will write my question. Read. Considering the variety of molecules involved in cell death mechanisms such as apoptosis, how are the targets selected theoretically? Have bioinformatic techniques replaced deductive or inductive speculations? Deductive is if you analyze the cascade, we can imagine one target, and inductive if one target that work in some other can also work in another. And how you select the targets?
Or first of all, you know, multiple ways. One is you look at the human patient samples and you see what are the levels of expression of the protein, and especially if you can look at the activated form. For caspases, maybe cleaved form, for RIP1, RIP3, phosphorylated form, and so forth. So you look, is there a relevance from the human samples? The other one is, can you target this protein? Does it have enzymatic activity? Does it have a protein binding site? Does it have some epitope that you can target with other antibody if it's a receptor, or with small molecule if it's intracellular protein? And third, you look at the safety. You can make a genetic models like I showed you for RIP3 and RIP1, right? Knockouts don't tell you always everything. Only when you mutate the region that you want to target, you will know really how your compound or reagent will react. So that's why we learned that if you mutate kinase activity of RIP3, just one amino acid mutation causes lethality of the mouse, you know? So that definitely eliminates it from our consideration. For RIP1, it didn't. It validated that it's a good target. So we use all these factors in consideration: human data, targetability, druggability, and then biological validation from in vivo models.
You talk about some kind of paradox, and you imagine or you have really know about the potential of bioinformatics to get around this kind of paradox?
You know, bioinformatics or AI, whatever you want to call it, can help you with looking at the levels of expression because data may be available. You can use AlphaFold or some other predictive tools to see what is druggable or not. But safety, you cannot predict until you do your in vivo experiments. That's one thing you cannot predict, I think.
Thank you.
At least so far.
Thank you.
Mm-hmm.
Okay, guys, I think we are already done. Thank Domagoj so much. We have experienced trouble with Doug's audio and video. We will try to figure this out in a minute. So Domagoj, super thanks again. It's been real excellent to have you here in the course. Okay, and as I told you in the beginning, hope that we can see each other face to face very soon.
Absolutely. And if there are some burning questions, you can.
Oh, here's Doug! Yay!
Oh, there he is. All right.
All right.
Thank
Doug.
you, Domagoj. Send my love to
Well, and thank you all for your attention, and hope to see you in person one day.
Thank you very much. All right.
And I'll see Peter in the fall or sometimes. Okay.
Yeah.
And Doug probably in July.
Yes, I hope so, Domagoj.
All right. Take care. Bye.
Bye.
Bye.